Okay, so for our next set of topics here, we're going to turn our focus to the base of the biological hierarchy, molecules down here. In some ways, this portion of the course is going to feel like a crash course in chemistry because there are a number of chemical principles that determine how living cells and organisms work. And one of the amazing things about living organisms is that there's a dazzling amount of complexity to the biochemistry of any one living thing. So to give you a sense of this, here's a thumbnail view of a chart that outlines all of the metabolic pathways that are found inside of cells. And this entire chart, plus a companion chart that is organized according to cellular and molecular processes, they're available at this link on the slide in case you're curious about exploring them further. But you know, as you'll see here, this is a pretty overwhelming level of biological complexity. And a lot of this stuff is going to reappear in cell biology for those who are continuing in the biology major. But in any case, at this point, this chart's a little overwhelming. So the question is, how can we get started with understanding the biochemistry of living things? Well, I think one answer comes from an older biology textbook. It's actually a favorite textbook of mine, as seen in this quote here. So as it says, it's been said that it's necessary only to be able to recognize about 30 molecules for working knowledge of the biochemistry of cells. And yeah, it might seem like 30 is still a fairly large number, but we can group those molecules into categories as well and tackle things one by one. And if we do so, here's what we come up with. So we come up with water, 2 types of sugars, glucose and ribose, 1 fatty acid, a type of lipid, the phospholipids, 20 amino acids, and then 5 different types of nitrogenous bases. And note, as I say on this slide, for this course, you're not going to need to memorize the exact structure and function of all 30 of these molecules. However, there are a couple of exceptions to this, where by learning a specific set of structures and functional groups, that's going to help you be able to spot the different types of molecules in the wild. And from there, you're going to be able to deduce things about how things work in specific contexts. And I'd also note that if you're going to continue in your study of biology, I actually think it will be helpful to know these compounds by memory. But rather than brute force memorization, I suggest putting the emphasis on understanding similarities, differences, and unique features of each type of molecule. So now, here's how we're going to break up our studies of biochemistry. First, we're going to do a bit of a chemistry crash course and talk about how elements bond together to form molecules and why that matters for living things. Then we're going to talk more specifically about some unique and emergent properties of water, and specifically the things that have made this watery planet Earth a suitable place for life to arise. Then we're going to turn our attention to the carbon-based macromolecules. We're first going to talk about the properties of carbon that enable it to function as a backbone for macromolecules, and then we're going to explore the 4 major types of macromolecules found in living organisms and how they're combined to give rise to the more complex structures in cells. As I go through this part of the course, I also would like to note that if, like me, your previous chemistry background from high school was pretty weak, you may discover that you need to spend more time learning about and practicing some of the concepts that I'm about to go over. And that is totally okay, but it's really important that you recognize this about yourself sooner rather than later, and that you take charge of learning about these basic chemical principles on your own or with a study buddy. And as you're doing that, consider your textbook to be one good resource for this purpose. You might also find it helpful to seek out some of the tutoring services that are available at Siena, or this is something that I'm always happy to discuss with you during office hours, and I can help direct you to some of those tutoring services as well. Okay, so let's now start by talking briefly about properties of the chemical elements. So the first question is, so here's our table of the elements. Which out of the 94 naturally occurring elements should we even focus on? Well, to help us think that through, here on this slide, I've got the full molecular formula for one human, so such as me eating this cookie here. So here's an entire human according to atoms. Conveniently enough, the 6 most abundant atoms in this human, so carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, are the most abundant elements that are found in most living organisms. So in general, we're going to be able to focus our efforts just on these 6. So the next question is, well, what do we actually need to know about these atoms? First, we need to know what atoms are made of to know more about how they function and interact with each other. So, in a nutshell, atoms are made out of groups of protons, neutrons, and electrons. And hopefully that's a familiar concept to you, right? The protons and the neutrons make up an atom's nucleus, and the total number of protons and neutrons are collectively going to determine an atom's atomic mass, so how heavy that atom is. And hopefully, as a reminder here, as the name suggests, so protons are positively charged and neutrons don't carry a charge. And it's now the number of protons specifically that determine which element we're working with, right? So number of protons. And at the same time, the number of neutrons can vary for a given element within reason. So for elements where there's variation in the number of neutrons in the nucleus, we refer to those variations as isotopes of that element. And to give you one example that crops up time and again in biology, the element carbon has three main isotopic forms. So most of the carbon that's present in the environment is carbon-12, and carbon-12 has 6 neutrons, so 6 neutrons, 6 protons, molecular weight of 12. But there's carbon-13 can also occur, and carbon-13 is actually a stable isotope of carbon. So in that case, it's going to have 7 neutrons rather than 6. And meanwhile, there are also trace amounts of carbon-14 kicking around, so 8 neutrons in that case. But unlike carbon-12 and carbon-13, carbon-14 is unstable, so it's considered to be a radioisotope. And carbon-14 will occasionally let fly with a subatomic beta particle from its nucleus. And actually, because carbon-14 is so rare, it can also be used as a tracer molecule to track the fates of specific carbon atoms in animals. And if you want to learn more about that, I'm happy to talk more about what it's like to make radioactive crickets, as shown here, in office hours at some point. So anyway, that's atomic identity. So it's the third building block of atoms that gives rise to the ways in which atoms interact, the electrons. So the nucleus of an atom is positively charged, containing protons and neutrons, and then the space surrounding the nucleus of an atom contains electrons. And individual electrons tend to repel each other, and so as a result, they're not found uniformly distributed around the outside of a nucleus. And instead they occupy defined regions that are referred to as orbitals. Along with this, elements are most stable when the number of electrons found in their orbitals are balanced. We're not going to go into the full details of this here, that's more of a chemistry topic, but it's important to note that an atom's chemical behavior is going to depend on the electrons present in the outermost orbital, which is also known as the atom's valence shell, right? So as I've said here in this slide. So until fairly recently, chemists had only been able to deduce that this was how atoms were built. But in very recent years, there have been a series of technological breakthroughs that have allowed us to actually directly visualize electrons in their orbitals around atoms. So that's what's shown over here in this image on the right. And the really amazing thing that you can see in this image is changes to the valence or outermost shell of these molecules depending on whether the molecule has a -1 charge, as shown here, so one more electron than a number of protons, or a +1 charge, as shown up here, so one fewer electron than the number of protons. And so anyway, I just think that this is a really amazing image. So in any case, if there are unpaired electrons in one or more orbitals in an atom's valence shell, that is going to cause the atom to be reactive. Basically, atoms will interact with each other in ways that are going to complete their valence shells. And the specific ways in which atoms handle their valence electrons determines what happens next, whether they wind up sharing or completely transferring over electrons, how evenly or unevenly those electrons are shared, and just how many electrons are involved in the exchange. So we're going to look at hydrogen for a quick example. So here's an electron distribution diagram for a number of the atoms that we're going to work with. So hydrogen up here has an atomic number of 1, so its nucleus consists of just a single proton. That also means that this hydrogen atom just has a single electron in its valence shell or outermost orbital. So this single electron is unpaired, and that means that that electron is going to want a buddy one way or another. So one way to do that would be to pair up one hydrogen atom with a second hydrogen atom and have them share both of those electrons. Okay, so here is my proton in the nucleus of hydrogen, and it's just got one electron in its orbital. Here's a second hydrogen atom. It's got one electron in its outermost orbital. So if we want to balance these out, if they share electrons, I'm just going to symbolize those electrons with a little dot here, then they're going to have a complete outermost orbital. In this scenario, there's another important thing going on here, which is that each of these atomic nuclei are identical to each other. So they each contain just a single proton. And what that means is that each nucleus is going to exert the same level of attractive force towards the electrons as the other nucleus. Or in other words, each nucleus in this case has the same electronegativity as the other nucleus. And so both nuclei are going to share the electrons equally. Okay, so now when electron or electrons are shared between atoms, the result is the formation of a covalent bond. Okay, but there's some additional complexity to this sharing, which is what happens when the atoms that are involved do not share electrons equally. So in other words, when one of the participants is more of a winner at the game of tug-of-war because it has a higher electronegativity. So now here's a chart showing atomic electronegativity across the periodic table. The main thing that I want you to notice here is that an atom's electronegativity is a product of two things. So it's a product of that atom's atomic number, so how many protons it has in its nucleus, right? So more protons is going to mean a stronger positive charge and a greater pull for electrons. And then the other factor that determines an atom's electronegativity is how far away the valence or the outermost electrons are from the charged nucleus. So I want you in this table to now focus your attention on one atom in particular, right here, that atom oxygen. So you'll notice that hydrogen up here has fairly strong electronegativity because it's got one electron in its valence shell. Its valence shell is as close as you can get to the nucleus. But hydrogen's electronegativity is weaker than the electronegativity of oxygen. So if we flip back to looking at oxygen's valence shell here, notice that the oxygen has two unpaired electrons in its outermost valence shell. So the first valence shell holds 2, second valence shell can hold 8. So two unpaired electrons here. So those two unpaired electrons plus strong electronegativity means that if this oxygen bumps into two hydrogen atoms with unpaired electrons, then all three atoms can be satisfied with full valence shells. But because oxygen has greater electronegativity, the electrons in this molecular dance party are going to spend more of their time in close proximity to the oxygen and less of their time hanging out with the hydrogen protons. Okay, so let's just draw a picture of that right here. So here is the oxygen nucleus. Here is a hydrogen or a proton and a hydrogen, and initially we've got just a single electron in the outermost valence shell there, and for this oxygen we've got two unpaired electrons. So if they share electrons, so if this is shared, So then now this hydrogen has 2 electrons in its outermost valence shell, so does this one. But the oxygen atom has a stronger electronegativity than the hydrogen atom does. So it's going to pull the electrons towards the oxygen nucleus and away from the hydrogen nucleus. Okay, so we've got 2 contrasting cases here. We've got hydrogen atoms over here who are team players, sharing electrons equally in one type of covalent bond. And meanwhile, we've got oxygen over here hogging all the electrons, causing its neighboring hydrogen atoms to do without. And now the net result for this molecule, and I hope you can recognize that this is a water molecule, is that there's now a partial negative charge surrounding the oxygen atom from the uneven distribution of electrons, and a partial positive charge by each of these hydrogens. And we symbolize that there are partial charges by using a lowercase delta symbol, like so. It's going to be delta positive, and by the oxygen we're going to say delta negative. And so when there's a partial charge present, we're going to refer to the resulting molecule as being a polar molecule. So this is another example of a covalent bond, but it is polar or charged because the electrons are not evenly distributed around these 2 nuclei. So in contrast, over here what we've got is a covalent bond that is non-polar, since the electrons are shared equally in this case. Okay, so that's not all we have to think about. There's a couple other configurations to consider. First, there's actually a different way for oxygen to get all the electrons at once, and that is if it teams up with a second oxygen atom. So down here, if we have one oxygen atom, 1, 2, 3, 4, 5, 6, and another oxygen atom. So in this case, what's happened are sharing of 2 electrons collectively. And so in that case, if both of those oxygen atoms are willing to share 2 pairs of electrons, they're going to be happy with those 8 electrons in their outermost valence shell. And a case where 2 electrons are getting shared is referred to as a double bond. Okay, and in addition, let's take a quick look back at carbon. Okay, so carbon here, you'll notice, has only 4 electrons in its valence shell, and this outermost valence shell would like to hold 8 electrons total. So what that means is that carbon can actually form up to 4 different bonds with other atoms. In the simplest case, let's say that carbon runs into some hydrogens, and then, perfect, if there are 4 hydrogens involved, everybody's happy again. There's a pair of electrons for each hydrogen, and there's 8 electrons total for carbon. Now, in this case, carbon's electronegativity here is not as strong as oxygen. And so carbon actually doesn't hog the electrons in the same way that oxygen did. And instead, what we're going to have here is another type of nonpolar molecule, methane. Okay, so here's our carbon. It's got 4 unpaired electrons. Here is 4 hydrogens with unpaired electrons. So hydrogen's electrons are going to be shared with carbon. But again, the carbon does not have that different an electronegativity from hydrogen. And so this resulting molecule, CH4 or methane, is also a nonpolar molecule. Okay, so for our last case for the moment, let's consider the situation for another pair of atoms. And so this pair is going to be sodium over here and chloride over here. In this case, sodium has a very weak electronegativity because if we go back up here, sodium has just 1 electron hanging out in its outermost valence shell, and that's in its 3rd valence shell, so it's much further from the nucleus, right? And then meanwhile, chloride over here has a fairly high electronegativity, and chloride is missing just one electron from its outermost valence shell. And so hopefully from this you can see where this is going. So in this case, chloride's electron pull is so strong that it basically just completely takes sodium's electron. And in this case, we refer to that type of result as an ionic bond. Okay, so sodium in its outermost valence shell has just one electron by itself. Chloride in its outermost valence shell has 7. And chloride has stronger electronegativity, so it's going to pop that electron off of sodium. So as a result, we're going to refer to this result as an ionic bond. So chloride is going to wind up with a full -1 charge, since it's still sodium's electron, and sodium, in contrast, is going to have a full positive charge. And now the thing that's going to keep sodium and chloride close to each other is simply that each of these has a charge, and the charge of this atom is opposite the charge of the other. So since sodium is +1, it could also wind up forming a similar type of bond if it bumped into another type of negative ion. Okay, so now we're going to sum this up in a single slide, and we're also going to talk about one last case. So here up top are some examples of covalent bonds where the electrons are shared. So again, they can be polar or they can be nonpolar. And there can be single bonds, double bonds, or in some cases also triple bonds. Meanwhile, ionic bonds happen when the electron is completely donated from one to the other. And then there's this final thing here in the lower right that I have labeled here as a hydrogen bond. So let's talk about what's going on here. Well, what's going on here is actually very similar to what happens with this ionic bond here, where there's full but opposite charges attracted to each other. So in that same fashion, when there are partial charges, opposite partial charges can also be attractive to each other. And this attraction is not as strong as the attraction when there is a full charge, but it can be close to the same strength. And so when this happens in water between a partially positive hydrogen and a partially negative oxygen, the result here is what is referred to as a hydrogen bond. And it's these hydrogen bonds that determine a lot of the properties of water and that are central to a lot of the biochemistry of living things. So we're going to talk more about those in the near future. So the final point that I want to emphasize in this video is that anytime there are changes in atomic arrangements, so any case where molecules form or break apart, there's going to be a transfer of energy that happens as well. So either energy is required to cause the transformation, or energy will be released. So as illustrated here. So a chemical reaction is a rearrangement of atoms that's going to involve the storage or release of energy. And we're going to talk about this concept as well further in an upcoming video.